Okay, so we're going to look at the first part of gene expression, which is called transcription. Transcription is the process that makes RNA, period, end of story. Transcription is the process, the way a cell makes RNA. And all RNA has to be a copy of some section of the DNA that's in the chromosome. So any section of DNA in the chromosome that is copied by transcription to make an RNA can be called a gene. And because there's a copying of DNA, but the copy is going to be made in RNA, it's going to involve a polymerase enzyme. This time, the enzyme that is involved is called RNA polymerase. So RNA polymerase makes RNA as the product. DNA polymerase made DNA as the product in the previous chapter. So RNA polymerase has many of the same rules as DNA polymerase, but RNA polymerase doesn't need a primer. And in this process, we're only copying a short section of a chromosome. And so there's only going to be one strand that acts as a template. And we're not going to be copying both strands for the process of transcription. So transcription is a much smaller scope. It is copying from the chromosome, but it's not copying the whole chromosome. It's not copying both strands of the chromosome. It's just copying one strand of one short section. And the product molecule will be a polynucleotide RNA strand. So it has some similarities with DNA replication, but we're going to try to focus on the differences. So the good news about transcription is there's really just one enzyme, RNA polymerase. And there are signals in the DNA, in the chromosome, that tell the RNA polymerase where the beginning of the gene is. And there are signals that tell where the end of the gene is. And so that helps the polymerase know where to start copying and where to stop. Because, like I said, this is not DNA replication, this is transcription. So we're just going to copy a short segment in this process. This is a very famous microscope photograph. What you can see here, let me get my pen, is the DNA molecule. I'm going to try to outline it here in red. Stretches across this image here. Actually, I think it goes down here. I'm not sure on this other side, but that's definitely the DNA strand, at least in part. And what you have in this image, what they were able to capture are RNA polymerase proteins, because these are enzymes, they're proteins, that are moving along this DNA and they're building RNA. In this image, you can't see the RNA so clearly, but we have some other micrographs in science that show the RNAs also in the image. But this one is primarily the only resolution you get is just seeing the RNA polymerase enzyme as it's moving along a DNA template. So I'm going to show you a video, a short animation, because for this process, I think an animation helps enormously. I could explain it in words, but an animation is really going to help us.
The synthesis of messenger RNA is called transcription. In bacteria, transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of RNA polymerase called a sigma factor participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it unwinds and opens a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
Okay, so what you saw there is a good example of the basic process of transcription. In that video, they were focusing on something called mRNA, but it could be any kind of RNA. So whatever the RNA is, and the way the RNAs are named really has to do with what their function will be later. And so when they're being built, they're all built by this same process. So the name of the enzyme is RNA polymerase, and they mentioned that sigma factor, which helps to bind the RNA polymerase to the starting point of the gene, which has a name. It's called the promoter. The promoter is a particular sequence of nucleotides in the chromosome, in the DNA, that attracts that polymerase to attach there, and then that's where the copying starts. Just in front of the promoter is where transcription actually starts. And in the video, they were showing the RNA strand that was being built in purple. And what you notice is that only one of the strands is being copied. The other strand of DNA is just kind of sitting there. So we call the strand that's being copied the template, similar to the name we used, or the same name we used for DNA replication. But the strand that's not being copied in transcription is called the non-template strand. So only one strand is copied in this process. And we're making a single-stranded RNA polynucleotide. It's being built from 5' to 3'. And when the RNA polymerase reaches the termination signal, you saw that was also a sequence of nucleotides in the DNA, in the chromosome, and that causes a hairpin to form in the mRNA or in the RNA, and then the RNA is then released. The RNA polymerase is also released, and so that's how you make that little RNA molecule. Now you have an RNA molecule at the end of the process. And so this can happen over and over again on that same gene, because what you noticed is when the gene opened up and that RNA polymerase copied it, when everything was done, you probably noticed the DNA came back together. And so the gene in the chromosome is not changed by this process. So multiple RNA polymerase molecules can load on at the promoter of that same gene, slide down, make a copy, and again you can get many copies of that gene, so many RNA molecules of the same type. So the promoter, as I said, is a particular sequence of nucleotides in the DNA, and it's in the front end of the gene. We usually call it upstream. And one promoter sequence is, we usually pronounce it just TATA, T-A-T-A. So thymine, adenine, thymine, adenine. So that sequence is in the promoter of every gene. There are some other nucleotide sequences that are found commonly in genes, but we know that these particular sequences must be important because we see them in front of every gene. We know that must have a function in helping that RNA polymerase to attach there. And then the RNA polymerase simply goes along, and whatever it sees in the template, it adds the complementary nucleotide in the new strand of RNA, and then it keeps moving along. And when it gets to the termination sequence, then the RNA polymerase and the new RNA polynucleotide strand, which we call the primary transcript, releases from the DNA template and release from each other. And so that RNA polymerase can go back to the promoter and start over and make another copy, which often it does. So all kinds of RNAs are made by transcription. I had a list on the previous lecture segment of a short list. It's not a complete list actually, but there's lots of types of RNAs. When I was a student, they told us that there was only rRNA, tRNA, and mRNA. That was it. But since then, and I think y'all have a sense of this at this point, that's what they knew then. And so they taught us what they knew, but we've learned more since then. So now we know there are lots of different RNAs. And what we mean by different RNAs is that there are RNAs that serve different functions in the cell. They're all made of RNA nucleotides. They're all polynucleotide strands, but they serve different purposes or functions in the cell. So all kinds of RNAs are made by transcription, and all genes start with a promoter and end with some kind of terminator, and that signals how the RNA polymerase knows where to start and where to stop copying to make that RNA. So that's what a gene is. In the DNA, in the chromosome, a gene is a segment of the chromosome that is copied by transcription to make an RNA. But only mRNA goes on to the next process called translation. So what I'm saying is for some types of RNA, gene expression is just transcription. Sometimes there's some modifications to the RNA after transcription, but fundamentally, transcription for some genes is gene expression because the RNA that's produced goes immediately on to do a job in the cell. So RNAs can do jobs in the cell. In fact, there are some RNAs that are enzymes. When we think of enzymes, we usually think of proteins, but more and more we're discovering enzymes made of RNA. But in any case, for the mRNA, that's the one that's going to go on to the second process called translation. And you have these supportive RNAs called rRNA and tRNA that are going to be also, I would say, assisting in translation, or they have some role in translation. So these are the RNAs we'll be talking about in the next segment. But before we go on to translation, I'm going to go over mRNA modifications. So once an mRNA is made, it's called the primary transcript or the pre-mRNA. This is only for mRNA. And so this is also, modifications only happen in eukaryotes. Prokaryotes, none of the RNAs are modified. What do we mean by modified? Just like when we talked about what happens to proteins when they go through the Golgi and they get modified, RNAs, when they get modified, is similar. It's either something is attached on, something is trimmed off, something like those are modifications. Adding something, covalently attaching things, could be functional groups, could be, or removing something, trimming something. And so in the case of mRNA modification, what's going to be added is a special guanosine nucleotide at the 5' end of the new mRNA, and we call that the 5' cap. And it's believed that that helps the mRNA bind to the ribosome and protects the mRNA from being destroyed in the cytosol. And then there's also an enzyme which will add a long chain of adenine nucleotides on the 3' end of the mRNA. And that's called the 3' poly A tail. In this diagram, they're just showing maybe 7, but think of it more like 100. So like 100 adenine nucleotides will be built onto the end of every mRNA in eukaryotes. And then there's this third step, which is really interesting, the removal of non-coding sequences called introns, called splicing. So splicing is all genes have instructions for not only for making an RNA, but then for the mRNAs for making a protein. And so what's interesting in eukaryotes is that the gene contains the instructions and some junk sequences alternating along that chromosome. So in the chromosome, there's a start point for transcription called the promoter. There's an end point called the terminator. But in between that, intermittently, you have these junk DNA sequences that are not needed for making the final product. And so those are all copied into the mRNA. And so you can see in this diagram here, this represents the gene as it lies in the chromosome, in the DNA. When it's copied into the mRNA for the primary transcript, everything is still there, plus the cap and the tail. But there are sections called introns here. I indicates intron in this diagram. And introns are non-coding, or I think what's more helpful is interrupting sequences. They're interruptions in the sequences that are important. And so introns have to be removed, which is a little counterintuitive. Introns have to be taken out before the mRNA is ready. So the sections that have instructions are called exons. The reason they're called exons is those are the sequences that need to be expressed. Those have the important information in them. So the exons have to be left in, and the introns, the interrupting sections, have to be removed or taken out. So you can see how they're like alternating here. And you can have an exon of any length followed by an intron of any length. There aren't rules as to how long these are. Typically exons are shorter than introns. So by the time all the introns are removed, the final processed mRNA will be much shorter than the primary mRNA. So the mRNA, when it's first made, is quite long, but once all the introns are removed and the exons are then spliced together, and you get what we call the mature mRNA, that's going to be quite a bit shorter, typically. And there is a little machine in the cell that does the splicing. It's called the spliceosome. And the spliceosome consists of proteins and a special type of RNA called small nuclear ribonucleic acid. And that makes snRNPs. Anyway, it's all part of this structure called the spliceosome, which does the splicing. It removes the introns and it connects the exons together. At that point, the mRNA is called mature. So before there's any processing, it's called primary or pre-mRNA. And then you add the cap, the tail, and splice out the introns, and then the mRNA is mature. I didn't choose the terminology here, but that's the language that we use. All right. Now it turns out that the way that in eukaryotes, and in this slide focusing on human, but really it's true in eukaryotes, the splicing can happen in a couple different ways. In other words, many primary mRNAs can be made from the same gene. So copy after copy after copy from that same gene. But what can happen is in some of those copies, the primary transcript can be spliced slightly differently because it's possible not only to splice out the introns, but to also splice out some of the exons. And so all the introns are always removed, but in some cases, some of the exons are removed. And you could have from one gene, you can have exons that are removed in different combinations such that one gene, which has, several exons and introns, can be spliced together into different mature mRNAs. So let me draw a picture here. Let's say let me go back to this slide. Let's say you had in this transcript, there are 4 exons and 4 introns in the primary transcript. So what can happen is, after splicing, it's possible to get a final mature RNA. Sorry, I don't know why it did that. You can get a mature mRNA that has, for example, exon 1. I'm just going to draw it like this, exon 2, exon 3, and exon 4. But you could also get a version of that which only has exon 1, 2, and 4. So exon 3 has not been included in this particular mRNA. And so you can have these different versions of mature transcripts that include or don't include some of the exons, and that gives the cell the ability to make, I would call it, slightly different versions. We call them isoforms of the same transcript. Slightly different forms are possible from the same gene because each of those exons can be left in or left out of the final transcript. So it gives some flexibility that the genes can be used. It says this explains in part how 20,000 genes in the human genome can somehow encode 85,000 slightly different, at least, mRNAs. And that gives you more variety of proteins in the cell. All right. So when I was a student, alternative splicing was a brand new concept. Somebody had just found one example of it, and I was taught that this was very rare, very unusual, but it turns out probably most genes have this characteristic. So it's actually, in 2024, we say this is very common. This happens a lot. A lot of genes exhibit this. So again, making a point that when you're learning something, we're only teaching you what science believes to be, the data shows right now. But there's always ongoing research, even in these very basic, in fact, lots of research still in these very basic cellular processes. All right, so now we're done talking about transcription and then also about the mRNA processing that goes on in eukaryotes. The next section will be on the second part of gene expression, which is called translation.